Thank you so much for joining us for our two-part workshop with the Yale Child Study Center. And today we're going to be exploring the genetic side of autism. And I'm really looking forward to this presentation. And we want to thank Dr. Roger Joe and Dr. Abha Gupta from Yale Child Study Center. We've had a long partnership with the center. We've been very fortunate to be here in Westville, just maybe 15 minutes away from Yale. Yale Child Study helped us to create our Asperger program here in New Haven. And Dr. Fred Volkmar is on our board. Dr. Ami Klin was also quite instrumental in helping us at Chapel Haven in developing programming. So we are very grateful for this partnership. Oh, and by the way, my name is Catherine Sullivan DiCarlo, and I'm Vice President of Admissions and Marketing at Chapel Haven. I've been here about 12 years. Obviously, really like it here to have stayed this long, and every day is very rewarding in this field. So I'm going to just give you a quick overview about Chapel Haven and do some introductions, and then we're going to have Dr. Gupta give her presentation, and you are welcome to put some questions in the chat function as we go along. And Dr. Gupta is happy to answer questions as she goes along as well. And finally, just a note that we are recording this session. We do get people who can't make it for things and really enjoy the fact that we can send them a recording. So what about Chapel Haven? Our name is Chapel Haven Schleifer Center, which is a mouthful, but we have been in this field now since 1972. And so we're approaching 50 years of serving adults with a wide variety of profiles. And our mission is to help individuals learn how to live independently. So we do have our founding campus here in New Haven, which we recently overhauled to the tune of about $40 million. So it's a beautiful new campus. And we also have a satellite campus in Tucson, Arizona called Chapel Haven West. And the kinds of profiles we serve, certainly autism is a big part of our program. Adults with autism, adults with Down syndrome, intellectual disabilities, cerebral palsy, executive functioning challenges, social communication disorders. So those are just some of the different types of profiles that we have tremendous experience with. And upcoming events. So today is part one of our series. Next Wednesday, we will have a talk with Dr. Roger Joe. And Dr. Joe is well known to a lot of our adults. He's done some one-on-one counseling here. He's going to be talking about medication as it relates to the spectrum and mental health. So that should be a really good session. If you haven't signed up, we encourage you to join us. And then if you're interested in learning more about Chapel Haven, we have a variety of programs and services. We also have Chapel Haven Online, a virtual program, which is a great way to get involved here. You're invited to join us on Saturday, January 16th. For our virtual open house that starts at 11 a.m. So start your new year off right, looking at the path to adult independence. So now it's my great pleasure to introduce Dr. Gupta, and she is an assistant professor in the Department of Pediatrics and the Child Study Center at Yale, which is world-renowned, and Neuroscience at the Yale School of Medicine. She is a developmental-behavioral pediatrician who directs an autism diagnostic clinic for young children. She also directs a research program that investigates the genetics and neurobiology of autism. So she's deep into the science, especially regressive forms of autism, such as childhood disintegrative disorder and sex differences in autism. And along with Dr. Joe of the Yale Child Study Center, she leads the SPARK project at Yale, which is the largest nationwide multi-site effort to recruit 50,000 individuals with autism and their families for genetic studies. And I believe that along with her presentation, Dr. Gupta will talk about the SPARK study as well. So Dr. Gupta, thank you so much for joining us, and I'm going to turn this over to you.
Great, thank you very much for the introduction. And I'm going to share my slides here. All right. Can everyone see that and hear me okay? Yes? Great. All right, thank you for inviting me to talk about the genetics of autism. This is a field that I've committed my research career to, so I like sharing this interest. I know that there are probably some of you listening to this who may not have been thinking about genetics or haven't thought about it since maybe even high school or college. So I thought I'd start off first with a couple of slides on Genetics 101, just to get everybody on the same page. So this is to remind you that the cells in our body contain a compartment called the nucleus. And the nucleus is where our genetic information is housed. And it's stored in 23 pairs of chromosomes. So we inherit one pair from our mother, one pair from our father. And here's a diagram showing these chromosomes. And so what makes up the chromosomal material is this DNA strand. You've probably heard of the term DNA double helix or DNA ladder. So this is kind of what it looks like in a very simplified form. But the most important part of this DNA strand are these rungs of the DNA ladder, which are called bases or nucleotides. And the reason why they're so important is because they pair up in very specific ways to make these rungs of the ladder. And the sequence of the rungs is what contains the genetic code, or what we call the genetic information that translates the DNA information into proteins. So the sequence of these bases, these rungs of the ladder, is what determines the sequence of amino acids in our proteins. All right. So here's another diagram showing a cell with its nucleus, which houses, again, the DNA double helix. It undergoes a process called transcription, where the code, the genetic code in the DNA, is copied into something called messenger RNA. And you've probably heard of this term, mRNA, in the news lately, because the recent vaccines that have come forward for coronavirus are known as mRNA vaccines. So now you know where that term comes from. They contain this molecule called mRNA. So this molecule is transported out into the rest of the cell, known as the cytoplasm, and it hooks up with the protein-making machinery of our cell. And so, as I mentioned, the sequence of these bases, these nucleotides, is translated into a string of amino acids that makes up proteins. And then you know proteins make up the rest of our body and all the functions of our body. So they're very important. All right, so what do human geneticists do when they're looking for mutations associated with a condition? So here again is a simple diagram of that DNA ladder and the rungs made up of these base pairs called adenine and thymine and guanine and cytosine. As I said, they pair up in very specific ways. So what happens is, let's say one of these rungs undergoes a change. And here's an example. Like, let's say this adenine changes into another, like a cytosine, for example. So what does that mean? Well, this, first of all, how does it happen? This type of change or genetic mutation can be spontaneous. It could happen because it was triggered by some kind of environmental factor like radiation exposure, or it could be inherited. Now, this change could be completely harmless. It might mean nothing for the protein that is coded here, or it could be harmful. It could cause a disease or a disorder. So human geneticists, when they're doing analyzing a patient's DNA, they're looking for these types of changes, and specifically those changes that could be associated with the condition they're studying. Because again, a lot of these changes can be completely harmless. So they have to look at the ones that might be associated with the condition they're studying. But, you know, sequence changes, so changes to those rungs of the DNA ladder, aren't the only type of mutation that geneticists look for. They also look for another type called copy number variants, or CNVs. And so, you know, we think about having, you know, again, two pairs of every gene, one inherited maternally, the other one inherited paternally, but it's actually more complicated than that. Sometimes we can have 3 copies of the same gene, or 1 copy of a gene. And so here's an example of a duplication where you have a chromosome, and for some reason, this part of the chromosome has been duplicated. So again, this could be spontaneous, it could be inherited, or it could be caused by an environmental factor. But the point is that there might be very important genes here where if you have too much of that gene, if you have an overdose, that could cause a condition. Here's an example of a deletion. So you have a chromosome in which you've lost this part of the chromosome, it's broken away. And even though the chromosome comes back together again, you've lost a chunk of material that could have a really important gene. Again, this could be spontaneous, it could be inherited, or it could be caused by an environmental trigger. So human geneticists not only look for sequence mutations, they also look for these copy number mutations or copy number variants. All right, so what's the evidence for a genetic contribution to autism? Well, it actually has among the strongest evidence for genetics among all complex neuropsychiatric disorders. So human geneticists calculate something called a heritability factor, which is how much of the clinical features of a disorder are due possibly to genetics. And for autism, it's quite high. It's about 80% according to recent studies. So other neurodevelopmental or neuropsychiatric disorders like bipolar or schizophrenia or depression have heritability factors in the 60% or 50-60% range. So autism is quite high. And so how do human geneticists come up with these kinds of numbers? Well, they do family studies. One type of family study they do is called a sibling recurrence risk analysis. So they look to see in families who already have a child with autism, what's the risk for a younger sibling to also have a diagnosis on the spectrum? And it's quite high, it's almost 20%, which is again much higher than the general population prevalence of autism, which is about 1 to 2%. But someone could argue, well, siblings grow up in roughly the same environment. How do you know that it wasn't an environmental exposure that caused autism rather than, a genetic mutation? So human geneticists try to get around that by doing something called twin studies. So what they're doing here is they're looking at identical twins and fraternal twins and seeing how often do they share a diagnosis within each twin pair. So identical twins, share 100% of their genetic information. And fraternal twins are like any brother-sister pair. They share about half their genetic information. And so these studies have found that identical twins share a diagnosis on the spectrum much more frequently than fraternal twins. So if you're assuming that each twin pair is, growing up in roughly the same environment, if identical twins share a diagnosis on the spectrum more frequently, then you think something genetic is going on, right? Because they share so much more of their genetic information. However, the important thing is that identical twins don't always share an autism diagnosis. There are plenty of examples of identical twins in which one twin has autism and either other one doesn't. So they're known as discordant twin pairs. And what does that mean? Well, it tells you that it's not 100% genetic, that there are probably a combination of genetic and environmental factors that cause autism. And so an interesting tidbit for the day is that, there are about 20,000 some genes in the human genome. About 80% of those are active in the brain. So that's a lot of genes to try to sort through to figure out which ones might be contributing to autism. But imagine how many millions of possible environmental factors there might be out there. And it's much harder to parse through those. And they haven't received as much systematic investigation as the genetics has. So anyway, any human geneticist in the field would tell you that it's a combination of the two, and we have a lot to uncover yet. And another final line of evidence for a genetic contribution is the fact that about 10% of cases, some studies say even more, have an overlap with a known genetic syndrome. So here's a table that shows some of those autism-related genetic syndromes, and some of them might be familiar to you, like Fragile X syndrome or Rett syndrome, tuberous sclerosis. And so some of these have been traced back to very specific mutations in specific genes, which is helpful to us because if you have a disorder like Fragile X, that can also result in autistic features in an individual, knowing that the syndrome is caused by mutations in a gene called FMR1 helps us, it might shed light for us on the neurobiology and the genetics of autism as well. So if you look at the proportion of patients who have this syndrome that also have autism, for Fragile X, there's quite an overlap. About a quarter of males with Fragile X also meet criteria for autism, about 6% of females. The important thing, though, is that if you look at the reverse, so what proportion of patients with autism have that syndrome, it's quite low. meaning that, although these syndromes might shed light for us on the neurobiology of autism, none of them explains a big proportion of cases of autism. They're rare causes of autism, like on the order of 1 to 2%. But still, they're still valuable to us in trying to understand the neurobiology of autism. Okay, so I want to present the bottom line up front, which is that autism spectrum disorders are complex. There's wide phenotypic variability. There's a wide spectrum of features. There are likely multiple etiological factors. There's a great deal of genetic heterogeneity. And what I want you to take away is that there is no gene for autism, not even a few. In fact, scientists in our field believe that there are hundreds of genes that are responsible for autism. And that's why it's so complicated and why we're still in business and we haven't figured it all out yet, but we're hoping to. This make the point, like a schematic of all the human chromosomes. And so the tick marks on the left are showing chromosomal regions that have been highlighted by some type of genetic study in autism, showing that that region is associated by either, the finding of a chromosomal abnormality or a sequencing study or a copy number variant of some sort. So as you can see from this, there have been, chromosomal regions throughout the entire human genome that have been highlighted by some type of study in autism, meaning just again driving home the point that it's very genetically heterogeneous, that there are a lot of genes involved, a lot of chromosomal regions involved. All right, so how do human geneticists go about trying to figure out what parts of the genome might be associated with autism? So I wanted to give you a couple of examples. One is cytogenetic analysis. So this is a way that human geneticists look for chromosomal abnormalities in patients with autism. There's two main types of chromosomal abnormalities. One is balanced. Examples include inversions and translocations. And here's a diagram on the left showing a balanced chromosomal abnormality. So for example, let's say you have two chromosomes, A and B, and for whatever reason, they switch ends. Maybe again, it's spontaneous or it's inherited from a parent or it's due to an environmental trigger. So even though you haven't lost or gained any net chromosomal material, now you have two breaks, and those breaks could affect a really important gene that's active in the brain that might cause something like autism. So we look for balanced translocations being associated with autism. Here's an example of an unbalanced chromosomal abnormality. So everyone is pretty familiar with Down syndrome, which is caused by a whole extra copy of chromosome 21. And that has a lot of clinical implications. So that's an example of an unbalanced abnormality where you have extra chromosomal material. All right, so I wanted to show you a real life case that came to our lab that we tried to understand from a genetic viewpoint. So we got this diagram, this pedigree of a family in which you have unaffected grandparents who had a son who also is unaffected, has a normal complement of chromosomes, 46 chromosomes, X and Y. His wife, also normal, 46 with 2X chromosomes. They had 3 children, one of whom, their son here, shaded in black, was diagnosed with autism, and he was the only one in his family also that had an abnormal chromosomal finding, and that was a chromosome 7 inversion, which I'll show you in the next slide. So, the hypothesis becomes that if he's the only one in his family who has autism and the only one who has a chromosomal finding, then probably the two would be related, that the chromosomal abnormality might be contributing to his autism. But, we have to do some follow-up studies to really confirm that. So what we did was we took his chromosomes, we stained them with a dye. And so this pattern of dyes across the chromosomes can let us compare the pairs of chromosomes and see if there's any abnormalities. And so we have a cytogenetic expert in our lab who took a look at his chromosomes under a microscope. So this picture was taken from a microscope with his chromosomes on a glass slide, and found that one of his chromosome 7s was abnormal. So here's a blow up. And I'm not sure if you can appreciate it or not, but there's a different staining pattern to these 2 chromosomes. And it turns out, so one of his chromosome 7s was normal, but the other one had this inversion where a piece of his chromosome between here and here flipped upside down. And so again, he didn't lose or gain any material. chromosomal material, but now he has 2 breakpoints. And it turns out that one of those breaks went right through a really important gene that is active in the brain that's involved in nerve cell signaling. So that tells us that, wow, that might really be causing this child's autism. I want to tell you that this investigation, one of the very early ones we did in our lab, took about a couple of years to do. And so, you know, we were working on one family at a time, trying to find one gene at a time. It took a lot of work. It was a very labor intensive. But since then, our field has really been revolutionized by something called high throughput sequencing. And here's an example called whole exome sequencing. So what happens now is, you know, instead of looking at one family at a time, one patient at a time, what we can do is we can actually obtain the genetic sequence for all the genes in the human genome in one experiment. And not only for all the genes for one person, but for multiple people in one experiment. So the human genome has 3 billion of those base pairs, those rungs of the DNA ladder. And about 1%, actually only just about 1% of those rungs actually make up genes, which is what codes for proteins. The other stuff, the 99% of the human genome, which doesn't, are considered regions in between genes that for a long time we didn't know what they were doing, although they might still be important in how genes are activated. But anyway, so whole exome sequencing is obtaining the DNA sequence for all of those coding regions, which is known as the exome, all those gene regions. So here is a diagram of what this process looks like and why it's so powerful. So here's this fancy glass slide on the upper left where they have these 8 channels. And so you input into the channel the patient's DNA that we can extract from a saliva sample or from a blood sample. And then you can do this for multiple people on one glass slide. And so here's what's going on in those channels. And so the power of the technology comes from the fact that you can make hundreds of millions of copies of fragments of that patient's DNA. And that allows for really accurate sequencing so that if we're looking for even very rare mutations, we can detect them. And this is all done in parallel. So again, you're not looking at one gene at a time, you're making these clusters, these copies of every gene in that patient's genetic material. And so this is what the machine looks like, where you put in this glass slide and it spits out your data. And so it sits on top of a laboratory bench, and there's over a dozen of these in our genomic facility at the Yale West Campus. So what happens when we get the raw data back? So what happens is we get short sequences of DNA for this patient. And as I said, it's multiplied so many times that we get many copies of that DNA sequence. So we have a reference genome, which was produced by the Human Genome Project about 20 years ago, where the entire human genome was sequenced. And then what happens for us is that it becomes a bioinformatics challenge. Basically, we take these raw DNA sequencing reads, and then we try to line them up to the human reference genome and then look for changes. That's all we're doing basically, is looking to see where there might be a mutation, a sequence mutation like I described before. So we get these raw reads, we line them up, and then we look for possible variants, possible mutations. And so the challenge comes from the bioinformatics aspect of it, of analyzing all this data. And for just one person, when we do this whole exome sequencing, we can get nearly 100 million of these short DNA reads. And so for one person, we could get 30,000 different mutations across the entire genome. The vast majority of those are going to be harmless. So it's our challenge to try to figure out which of these might be associated with a condition that we're studying. So even if you wanted to filter out, let's say, look for just new mutations in a person, even that, is nearly 800 in this one person. So that's where our challenge comes from, is then we ask questions like, well, is this mutation in a gene that, is active in the brain, where in the brain, when in the brain? We ask all those types of questions to try to figure out if of those 30,000 some variants that we find in one person, which one or a few might be associated with autism. So that's where the challenge comes from, analyzing this type of data. All right, so why, one of the best questions I get from parents of children with autism is why do all this? Like, why do genetics testing? How will it help their child, right? Well, there's a number of different reasons to do this type of testing. First of all, you might be able to identify a known genetic syndrome like Fragile X or tuberous sclerosis that has a lot of overlap with autism. And the reason that's important is because some of these syndromes have additional organ involvement. So for example, Fragile X is associated with connective tissue problems. Tuberous sclerosis can affect other organs like the heart, kidney, and lung. So it's important to be aware of those. And it really allows for, anticipatory guidance for families of what they can expect for their child. Also, it has implications for genetic counseling. So here are two pedigrees, for example. Here's a family in which the shaded circles and squares means that there are multiply affected family members. Something is running through the family, right? Being passed on from generation to generation. Whereas you might have a family here in which there's only one affected individual, like it would probably due to something like a spontaneous mutation that wasn't inherited, but that occurred spontaneously. So that has a lot of different implications for genetic counseling, because you might have parents who are worried about having another child and what are their risks for having autism. Or there might be uncles or aunts in the family who are wondering, is something being passed along? So having genetic testing can give you some data for genetic counseling purposes. It can also be helpful in obtaining needed services if you have a positive genetics test. And this can also help families avoid pursuing further diagnostic tests that might not be so necessary, trying to chase down what might be causing their child's condition. So, and the other thing I want to point out is that clinical genetics testing should be routine no matter what. It doesn't matter how severe a child is in terms of their symptoms. So there was a small study done once that showed whether a child has classical autism or back then a condition known as PDNOS, which was considered a milder form of autism. The rate of a positive genetics test was still about the same. So it shouldn't matter if the individual has severe autism or mild autism. Everyone deserves genetic testing. And so what genetic counselors have found is that without any identifiable etiology, they can still use empiric recurrence risk data. So they can say that for a full sibling of a child with autism, the risk of autism is about 3 to 10%. If the affected child is a girl, it's 7%, whereas if the affected child is a boy, the risk is about 4% for a younger sibling. And the reason for this is interesting. The thinking is that girls might have some protective factors that make it harder for them to develop autism. And this really comes from genetic studies that have found that girls with autism tend to have more damaging genetic mutations, meaning they need like a bigger hit to actually have autism. And if a family has multiply affected children, the number really just does shoot up to 33 to 50% risk for a younger child. All right, so the other good question that parents always ask me is, again, like, okay, so let's say we find a gene. It might help us with some genetic counseling issues, but I mean, what's the end goal, right? What's the purpose of all this? Well, really, it's to find treatments. So I'm going to give the example of Rett syndrome, which is caused by mutations in a gene called MECP2, which causes most cases of Rett syndrome. So experts in the field have found that patients that show abnormal neurons, but not neuronal death. So the question is, can viable but defective neurons be repaired and reverse the symptoms of Rett syndrome? So a research group made a mouse model that lacked this MECP2 gene that causes Rett syndrome in humans, and found that the mouse model develops neurological symptoms just like the patients. What they did then was they genetically manipulated the mice so that they reactivated the gene in this experiment. But they did this in adult mice. They didn't do this prenatally or in baby mice. They did this in adult mice, and they were still able to reverse some of the neurological symptoms. So the big takeaway point here is that the absence of this gene doesn't irreversibly damage neurons. In fact, there is still time to intervene, even in later ages. And I think this just speaks to the plasticity of the human brain, and that we don't have to give up. We don't have to say, well, if you don't intervene in utero, there's nothing to be done. We should never be thinking that way. All right, so I know I'm running out of time, so I quickly just wanted to talk about SPARK, which stands for Simon's Powering Autism Research and Knowledge. And so I was talking about how, at one point we were just studying one family at a time, which has really been accelerated by having things like whole exome sequencing that has sped up our research. But Spark aims to speed up this research even further. So the goal of Spark is to recruit 50,000 families with autism, collect their clinical and genetic information through saliva samples. And then they are also committed to returning genetic results at no cost to participants, so it doesn't cost anything to participate, and hoping to group some of these families or some of these patients by genetic similarities and phenotypic traits to try to understand better how a genetic mutation might cause symptoms. And not only that, it's not just genetics that is the endpoint, it's also matching participants to other further research studies. So what Spark has been able to do is come up with this gene list so far that contains 178 genes and copy number variants. And I know it looks like a list of letters and numbers, but this is really important to us because these abbreviations for gene names gives us, a hint of, okay, which genes might be associated with autism, what kind of proteins do they code for, where and when in the brain are they active, and so what kind of, issues might they be causing at a cellular level. So that's why this is so important to us, to finding this gene list, because it helps us understand the neurobiology of autism. All right, and so families, and about 10% of families right now have a positive genetic finding, but we expect this to increase as we study more and more families and understand more about the genetics of autism. But families in which, an individual with autism has been found to have a mutation in an autism-associated gene, we'll get this kind of a report. So for example, this family, the individual had a mutation in a gene called FOXP1. So they get information about this gene, what it does, and what other patients with mutations in this gene have in terms of features of the disorder. And so that can be really valuable information to families. And as I mentioned, Spark participants can be involved voluntarily in many other research topics that might be of really practical interest to them. So for example, like sleep and diet, brain imaging, parenting stress, environment, all a number of different topics. I think there are some 80-some research studies out now going way beyond the genetics and looking at other types of topics that are important to families affected by autism. So who is eligible to participate? You must live in the US and be able to consent in English. Participants should be an independent adult with autism or the parent of a child or dependent adult with autism, and biological siblings are very welcome too. And the participants must have a professional diagnosis of autism to participate. Basically what they're doing here is they're submitting a saliva sample for genetics analysis. All right, so I just want to put this slide up and encourage you to join us in this big nationwide study that really hopes to accelerate research into the genetics of autism. You can visit our website, sparkforautism.org/Yale, or definitely please contact us at our email address or our phone number here up on the screen. And really, we appreciate family participation so much because that's what makes advancements in fields like the genetics of autism possible. It just wouldn't happen otherwise. So thank you very much. I'll take any questions anyone might have.
Does anyone have any questions? You can feel free to unmute and ask.
If you wanted to participate, we just contact you and we go down to Yale. You send us the containers to give you our saliva.
Yeah. Yeah, so definitely visit our website. It has all the information. It can be actually done entirely from home, where the Simons Foundation, which funds this project, can send the saliva collection tubes and instructions and prepaid envelopes, all to to return back free. And then we're also on standby. You don't have to come visit Yale to do this. You can do this entirely from home if you wish, but we can also do home visits if you're amenable to that. Not everyone is during the pandemic. It's up to you. Or we also have collection events that we can tell you about. But yeah, definitely visit the website or email us or call us, and we have a whole group of very dedicated study coordinators who can walk you through every single step of the project. The whole thing from beginning to end really should last about 40 to 45 minutes. So We try to make it as
And
easy as possible.
how long does it take to get the test results back?
It depends. There's whole exome sequencing involved, which can be a lengthy process, so it can take some months. I don't know what the exact time frame is now, and it depends on whether there is a result to report. But the important thing is that even if the first go around there's not a positive genetic finding, what Spark does is they reanalyze the data periodically, because we just gain more and more knowledge over time about what genes might be associated with autism. And so even if the first pass there's no known genetic mutation in a known autism associated gene, they will reanalyze the data periodically to see if a new more knowledge comes up about a gene and whether that mutation might be associated. So that's an important thing about this project going forward, is that they don't just stop at one point, they actually keep reanalyzing the data.
Okay. Now, I guess I would have to call and coordinate it, because I'm in New York and my daughter's in New Haven at Chapel Haven. Okay, sounds good. Thank you.
Yeah.
Dr. Gupta, we have a question actually from one of our teachers. Eric, did you want to ask your question?
Sure. It's not too in-depth, but I was just curious, when you were talking about PDNOS, is that now kind of an antiquated term? I wasn't sure if
Yeah, so so that was a part of the DSM-4, the Diagnostic and Statistical Manual of Mental Disorders. And so the DSM-5, the latest edition, came out in 2013, where we had these various sub-diagnoses before, like PDD-NOS and Asperger's and Rett syndrome, and it all became collapsed under this big one umbrella term called autism spectrum disorder in the DSM-5. Now, there was a lot of controversy behind that, and not everybody agreed with that. And there's still, now that it's been 7 years since the DSM-5 came out, there's talks again about like whether that was the right way to go or not. So the rationale was that, the thinking was that we don't know enough about autism on a biological level to make these like boundaries between various subdiagnoses like PDD-NOS and autism. And the thinking was it would be simpler for the field if we just called it all autism spectrum. But there's definitely a lot of people, and I'm kind of on this side, where I think there is so much, again, diversity in the clinical features that collapsing everything under one term, kind of you lose some of that information, which I think is important. So, there are definitely people out there, including researchers, my colleagues, who think that maybe we went too far in doing that and might kind of revert back to more some of these subdiagnoses. But again, it's not settled. There's, it's still controversial. I know in the DSM-5, there was a paragraph supposedly put in right before publication that said, well, if you have a well-established diagnosis from the DSM-4, you just get to keep it, which seemed kind of like a cop-out. But so it is a bit antiquated in terms of like, we don't use it so much anymore in terms of like clinical diagnosis in our clinics itself. We're not calling things people having PDD-NOS anymore, but some of us old timers still remember those terms, and feel that they still were valuable.
Well, in the public schools on the IEPs, a lot of times they will use PDD-NOS. So I was just curious about the trend, and working at a company, I want to be able to make sure that we're using the right up-to-date terminology and things like that, personally speaking.
To be completely up to date, the latest terminology it's all called autism spectrum these days. And I think unfortunately people are forgetting those other terms too, like especially the new trainees that we have up and coming in the field. They're not as familiar with them anymore. But who knows? I think it's still an open question. I think, maybe by the time the next DSM comes out, there might still be more changes, but who knows? It was like 20 years between DSM, so it might still be a while.
Thank you.
Thank you, Dr. Gupta. Mark, did you want to unmute and ask your question?
Sure. I think I'm unmuted.
Yeah.
Thanks so much. This is really terrific. And as a parent of an 8-year-old with an autism diagnosis, I have to agree that we're losing some of the granularity of the earlier DSM. In my experience, it's harder to tailor the kinds of things that can be done in the school to help with particular different kinds of autism. And I think that based on the experience of my own son's diagnosis, he probably would have been considered someone in the Asperger's category under
Yeah.
the old DSM, which is different from someone who's more along on the spectrum. So
Yeah.
that's really helpful. And you know, this is probably worthy of a whole session of itself. So it might be too open-ended a question, but how far off do you think we are from therapies that can actually mitigate you know, the impact of autism for young people?
Yeah, no, that's a great question. I think, you know, the research has just, I've seen it in my short time, like how much our work has been revolutionized by things like, you know, these new technologies like whole exome sequencing. It's just become routine now to do that, and it's really accelerated our work. I think already there are a number of drug trials that Dr. Joe can talk about even more in detail than I can, because he directs them at the Yale Child Study Center, looking at various you know, pharmaceutical agents, not just looking at, you know, helping to treat some of the, you know, side issues like that are important, like aggression or self-injury, but really the core features like the social issues in autism. And so there are a number of pharmaceutical compounds out there that people are really zoning in on, and they're really only possible because of the, you know, the neurobiology and the genetic studies that we're doing, because that identifies what cellular or molecular pathways might be amenable to pharmaceutical targets. So, I mean, it's obviously a big topic of research, a very hot topic in our field, looking for pathways in the brain that we can target with various compounds. And I'm not sure if Roger will talk about it at his next, do anyone know, Annie, or if he's going to touch on that, on the drug trials at the Child Study Center?
Sorry, can you hear? There's a printer behind me, sorry.
That's okay. No, there was a great, yeah, did you hear the question?
It was about the clinical trials, if Roger's
Yeah.
about in the next meeting. I'm not sure if he is, but we can definitely ask him to cover that, because that's part of the work we do here.
Yeah.
Yeah, we don't have anything currently ongoing, but definitely some few that will be up and running hopefully soon. So we can ask him to include those in the next talk.
Okay, when is the next talk, may I ask?
It is,
Sorry.
no problem.
Next Wednesday, right?
Next Wednesday, yep,
Oh,
at 12 p.m.
Great, okay,
Yeah.
great.
Yeah, he's the chief person at the Child Study Center who was you know, directing these drug trials.
Oh, wonderful.
Yeah.
Well, thank you. This has been terrific. I really appreciate it.
Oh, thank you for listening.
You might have one more message. Let's just look. Oh, yes, December 9th, which is next Wednesday at noon. All right. Well, Dr. Gupta, that was really fascinating. And I learned a whole lot in a very short time about
I could have gone on and on and on.
chromosomes and, well, you know, and that there's so many variations of the gene. I did not know that. So that
Yeah.
was really interesting. And so we thank you, and we thank everyone who joined us. And I hope that everybody can join us again next Wednesday. Dr. Joe will be fascinating as well. And he's very, very informed on this topic of medications, which I know that a lot of our families, you know, wonder about medication, and is my child on the right med? Should he be on a med, etc. So it'll be really illuminating. So thank you so much.
Thank you.